Welcome to this lesson on frameshift mutations. Frameshift mutations are a critical type of genetic mutation that can have severe consequences for protein synthesis. In this video, we'll explore what frameshift mutations are, how insertions and deletions cause them, and why they're so harmful to organisms. Understanding frameshift mutations is essential for grasping how even small changes in DNA can lead to major biological effects. A frameshift mutation is a type of genetic mutation that shifts the reading frame of the genetic code. This shift alters how codons are read during the translation process. To understand this, remember that DNA sequences are read as instructions to build proteins. When the reading frame shifts, it's like reading a sentence with all the spaces moved over by one letter. The entire meaning changes from that point forward, affecting all downstream codons and resulting in a completely different protein sequence. Before we dive deeper into frameshift mutations, let's review what a reading frame is. DNA and messenger RNA are read in groups of 3 nucleotides at a time. Each group of 3 is called a codon. For example, the sequence AUG pipe C, AU pipe GGC pipe, UAA shows 4 codons. Each codon codes for a specific amino acid or signals the end of translation. The reading frame determines which nucleotides are grouped together. If this frame shifts, completely different codons are formed. An insertion mutation occurs when one or more nucleotides are added into the DNA sequence. Let's look at an example. The normal sequence reads a UG pipe C, AU pipe GGC pipe, UAA now. If we insert a single nucleotide, specifically an A, after the C, the sequence becomes a UG pipe C, AA pipe, UGG pipe CU, a pipe A. Notice how the insertion of just one nucleotide has completely changed all the codons that come after it. The reading frame has shifted, creating entirely new triplet groupings from that point forward. A deletion mutation is the opposite of an insertion. In this case, one or more nucleotides are removed from the DNA sequence. Using our original example, a UG pipe C, AU pipe GGC pipe UAA, if we delete the A from the second codon, we're left with a UG pipe CU pipe GGC pipe, UAA. Just like with insertions, this single nucleotide deletion shifts the entire reading frame. All codons downstream of the deletion are now different, which will produce different amino acids during translation. Frameshift mutations have 2 major impacts on protein synthesis. First, they alter all codons downstream of the mutation. This means every single codon after the insertion or deletion is changed, producing a completely different sequence of amino acids. Second, they usually result in a non-functional protein. The altered sequence either produces a completely different protein that doesn't work properly, or more commonly, a truncated protein that's cut short. Either way, the protein typically cannot perform its intended biological function. Frameshift mutations are considered among the most harmful types of mutations for 3 key reasons. First, they change every amino acid after the mutation point, creating a domino effect throughout the entire protein. Second, they often introduce premature stop codons. These are signals that tell the cell to stop building the protein early, resulting in an incomplete, non-functional protein fragment. Third, the combination of these effects results in severe loss of protein function, which can lead to genetic disorders and diseases. Unlike point mutations that might only change one amino acid, frameshift mutations devastate the entire protein structure. Let's summarize what we've learned. Frameshift mutations are among the most severe types of mutations because they disrupt the entire reading frame downstream of the mutation. Whether caused by insertions or deletions of nucleotides, these mutations change every codon that follows, often leading to completely non-functional proteins. Understanding frameshift mutations helps us appreciate how precisely our genetic code must be maintained, and why even small errors in DNA replication can have devastating consequences for cellular function and organism health.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.